DNA fragmentation essay. So in this lecture, let me first talk about a very remarkable discovery which was made in 1997 from the Wang group. They have identified a DNA fragmentation factor which is also called as DFF. The link is been shown to you. You can visit to this link and then you can see that how this has been discovered. But essentially what they have done, as the title of this paper suggests from the Wang group, that DFF or DNA fragmentation factor, it is a heterodimeric protein. Now when they are saying it is a heterodimeric protein, it is basically components of two subunits. It consists of two subunits. What are those? We will see in the next slide. But what they are saying, that this is the function in downstreams of the caspase 3. The caspase 3 is a process which is involved in the apoptosis, and they are responsible for the DNA fragmentation. So I have taken one picture from this paper. Now what they are showing here, this is the lane 1, which is the caspase 3, okay, which is involved in your apoptosis or the DNA fragmentation. They have taken a HeLa S100 strain cells and they have taken a normal nuclear extract. When they have incubated the DNA, and if you see with the marker, this is the marker known, when they use just simple caspase 3 which has not been activated, because they have used by the way recombinant caspase 3, there is no fragmentation as you can see here. When they have incubated it with the HeLa S100 cell nuclear extract, there is not much fragmentation. And similarly, when they've used a normal extract, the plus sign indicates that they have added into the reaction, so there is no fragmentation. When they have used the S100 and caspase 3 cells, then you can see that there is a fragmentation. And when they use the normal extract and the caspase 3, they are not finding any DNA fragmentation. So DNA fragmentation is basically in a process of apoptosis. If I say this is a somewhere near about 200 nucleotides, small bands which is there, meaning say if this is your DNA molecules, there are certain endonucleases which will chop your DNA to make small fragments, and when you run a gel you will appear like this. So this was very remarkable discovery they have made, and they have identified that how it is working. A proposition which is also given, and I have taken from another paper. In this paper, this is a summarized diagram. As you can see, they are saying that the DNA fragmentation factor is consist of two subunits. Okay, so this is DNA fragmentation. It consists of DFF 45 and your DFF 40, as you can see in this diagram as well. So they are like in a complex. Now this complex is an inactive form. It requires its activity, and for this activity caspase 3 comes into picture. The caspase 3 not only separates these two subunits, but it also activates your DFF 40. So this DFF 40 is an endonuclease, and this endonuclease basically cleaves your DNA fragmentation. So it is like a DNase, you can say. The DFF has several other functions has been reported, that this is also work as a chaperone. Several mutants has been created by the same group, also the Wang, which I was saying, and they have shown that it is very much important for the developmental process. There are several other factors they have shown, like the high mobility group proteins and histones. They can also precipitate, and so probably they help in the process of the DFF 40 activity. So this is one of the endonuclease. Now this raises a question that do the cells have certain other endonucleases or not? The answer is yes to it. Okay, so these are certain endonucleases which is present, which can degrades the DNA, and this DNA degradation could be DNase 1 itself, which can degrade DNA in extracellular environment, the cyclophilins. There are certain cation dependent endonucleases which is involved in the process of T cell apoptosis. Apoptosis, by the way, is a process where we say it is a programmed cell death. This is of very much importance to the scientists. The reason for that is because it is involved in the process of the cancer. That is number one. The second thing which is involved here is that it is a process of a normal embryonic development, and the discovery of the DFF, which was also named later on as a caspase activated DNase, or in short it is called as CAD, is a very much contribution into this field, that how this process is been working. Now, with this introduction, let me talk about little bit the principle of the apoptosis. Okay, now the apoptosis is a term which is used to describe a controlled cell death, and this and most of the places it is used to eliminate the unwanted cell. It is different from your necrosis, which is morely associated with your inflammatory response, and apoptosis involve the cleavage of the chromosomal DNA into oligonucleosomal sizes. If you look into there are apoptosis process, it is caspase dependent, caspase independent, and this is usually taught in your cell biology classes. Okay, so if the apoptosis is somehow is not worked upon, means if it is not working properly, as I said, it is required for a normal development processes, then it leads to several human diseases like cancers, autoimmune diseases, and the neural degeneration. This is a just a repertoire of to show you that how this caspase dependent with the caspase 3 and the caspase 9 in the process of the embryonic stem cells. This is also involved, that is independent process, as I was mentioning. This is involved only one pathway which is involved. There is a, like that, for example, caspase 9, and in other way only caspase 3. So the caspase 3 and caspase 9, it is more or less, you can say, the effector endonucleases which work in the cellular system. Now, what are the morphological features of the apoptosis? So the apoptosis is involved in cell and nuclear, it is means if someone wants to see it, so it leads to cell and nuclear shrinkage. There is a chromatin condensation. In fact, your DFF, that is the DNA fragmentation factor, has also been shown in several papers to involved in the chromatin condensation. It leads to the formation of the apoptotic bodies, and then it is phagocytes by the neighboring cells. So it is a regulated process, and whatever the contents which is present, this content which cell uses for its own nutrients, because these apoptotic bodies has consist of DNA fragments or cellular components, they can be taken up by the neighboring cells, or it can be phagocytized. Now if we compare with the necrosis part here, so this is the two diagram which has been shown. Now this is a normal cell. If it is during the process of the apoptosis, as you can see here, there is a cell shrinkage, there is a membrane blaming, there is a nuclear fragmentation, there is a chromatin condensation, then formation of apoptotic bodies, and then these are taken by the phagocytes or the cells in the vicinity of that region. In the process of the necrosis, which also leads to the death of the cell, but here there is a cell swelling which is related with your inflammatory response, and there is a rupture of the plasma membrane, and then the post-lytic DNA fragmentation which take place. In apoptosis, the fragmentation takes place early, then the apoptotic bodies work. So this is the part 1 of a little bit theory about the apoptosis, because here the aim is not to talk about what the whole process of apoptosis, because this is not the theoretical lecture, but this is a practical lecture. So in my part 2, which I will be talking about the process or the procedure details, that how one can study